In our previous in vitro bioluminescence imaging video, we discussed the importance of characterizing stable reporter expression and light output levels in luciferase transfected cells prior to moving on to in vivo studies. Proper characterization of your cells in vitro is a critical step in successful in vivo experimental outcome. Before we begin the experiment, let's briefly discuss Revity's IVIS imaging system and the gas anesthesia setup. IVIS instruments are preclinical optical imagers with state-of-the-art optics, including a highly sensitive CCD camera located at the top of the instrument and cooled to -90 degrees Celsius to offer the highest sensitivity and eliminate background noise and dark currents. Additional features include multiple fields of view, depending on the number of animals to be scanned, an isoflurane anesthesia system with manifolds for mice and rats, and a heated stage to maintain the comfort of animals and prevent hypothermia. The first step is to inject the deluciferin substrate into our mice before we anesthetize them. Here, we used Revity's IVIS Bright pre-formulated ready-to-inject deluciferin solution. If using powdered luciferin, please dilute in PBS to a concentration of 30 mg per ml. Remember to sterile filter your solution prior to injection. Each mouse will receive an intraperitoneal injection of deluciferin at a final concentration of 150 mg per kilogram body weight. So, for a 20-gram mouse, the injection volume would be 100 microliters. While intraperitoneally is the most common route, a subcutaneous injection can also be used. After injection, luciferin circulates throughout the animal, eventually engaging with the luciferase enzyme in the target cells to produce light. Over time, the luminescence emission will increase, gradually reach a plateau, then decrease. It is highly recommended to determine when peak luminescence occurs, as this plateau represents the strongest, most stable signal. Mapping the kinetics of luciferin distribution for your model is imperative for absolute quantitative accuracy. Many factors will affect the shape of the kinetic curve, including animal temperature, timing of anesthesia, route of substrate administration, location of disease, stage of disease, etc. Deluciferin stays in circulation for about 2 hours before complete clearance. If you wish to perform repeat injections for short time intervals, wait for about 3 hours before re-injecting luciferin. As a precaution, please acquire a pre-injection image to determine if any trace light can be detected before injecting again. It is now time to anesthetize the animals for the study. We typically anesthetize animals using an isoflurane induction chamber that can be purchased as an optional accessory with your IVIS. The anesthesia assembly delivers a mixture of oxygen and isoflurane to the induction chamber. The gas mixture is also channeled into the IVIS to a manifold that splits the gas line into multiple ports so multiple animals can be anesthetized at once. There is also an active scavenging exhaust system consisting of a pump that routes excess isoflurane through a carbon filter. The filters need to be weighed often and the charcoal replaced according to the manufacturer's instructions.
Once you turn on the vacuum and oxygen source, choose your exhaust and oxygen flow rates, and select the percentage of isoflurane to use.
Usually starting at 1% and increasing as needed is a good rule of thumb, but please consult your veterinary staff and animal protocols for allowable doses. Breathing is a good visual cue to determine the accurate percentage of isoflurane to use. If the animals display labored breathing, drop the percentage. Once the animals have reached the desired depth of anesthesia, transfer them to the IVIS. If you smell isoflurane when you open the door, you may need to reduce the oxygen flow rate and/or increase the exhaust flow rate. Again, check the animals to ensure they're neither waking up nor displaying labored breathing.
As discussed, we injected our deluciferin substrate intraperitoneally. The substrate is available to cells almost immediately, but like any pharmacokinetic model, it takes time for the deluciferin substrate to be uniformly distributed to all tissues and cells in the animal. A measure of this uniform state is when the light intensity produced by the luciferase-luciferin reaction reaches a stable plateau. Prior to this plateau, the signal is highly variable. It is not recommended to image in this phase of luciferin kinetics because it will result in a high degree of data variability. Typically, the signal plateau post-IP injection is reached within about 15 minutes. It will stay stable for about 10 minutes and then decrease. We recommend that users image during this plateau phase to minimize data variability on account of insufficient substrate delivery to the target cells or tissue. This practice ensures that you obtain the most consistent and reproducible data when imaging at different time points during a longitudinal analysis. Metabolism and bioavailability of substrate can affect your curve dramatically. One of the most overlooked and underestimated factors is metabolism. Different animal strains may have varying kinetics due to fluctuating metabolic rates. Furthermore, animal handling procedures including the use of heating pads to keep the animals warm and timing of anesthesia can affect the kinetics. Please be consistent with your animal handling practice. As for our recommendations, we prefer to inject the substrate before the animals are anesthetized, and we recommend keeping the animals warm after anesthesia is administered. Bioavailability of the substrate will also impact kinetics and will depend on where the luciferase-expressing cells are located in the body. For example, a subcutaneous tumor growing on the flank might reach a stable signal plateau sooner than an orthotopic tumor growing in the brain. Other issues concerning tumor biology are the necrotic tissue and poor vascularization that develop as tumors grow. For very large tumors, it may take the signal longer to peak than was originally expected, as bioavailability of the substrate decreases when conditions within the tumor microenvironment deteriorate. It may be wise to repeat the kinetic analysis periodically if you want to study the progression of tumors over time to account for the shifting kinetics that can occur with tumor growth. Repeating this analysis can confirm if and when the kinetics shift. It will also ensure that your data is always acquired at peak bioluminescence signal output, even if that output shifts as a function of time. So how do we perform a kinetic analysis? Start by setting up a time sequence in the living image control panel. Using the sequence setup, we can direct the system to take multiple images with specific time delays between them. The kinetic analyses need not be performed on an entire cohort of animals. A few will suffice. In this video, we will use 2 animals bearing tumors in different locations. Start by opening the living image software, clicking Initialize, and setting the folder you wish to auto-save your data to. For more information on these steps, please see the in vitro bioluminescence imaging video. Select Sequence Setup in the IVIS Acquisition Control panel. Select the desired sensitivity settings. Using auto settings will prevent you from saturating your sources at later time points. To select auto settings, click the down arrow in the exposure window until it reads Auto. The default settings of medium binning and f-stop of 1 are recommended as a starting point. Auto settings will adjust the exposure time, the binning, and the f-stop as needed during the acquisition to attain the desired count values if possible. Select Add on the bottom right of the sequence editor window, and those image settings will be added to the sequence representing one image. We want the image acquisition to repeat for a defined number of times with a delay between each acquisition. It is recommended that you map your kinetics for approximately 45 minutes total and acquire an image every 2 to 5 minutes. For dimmer signals, we can extend the exposure time, and for brighter models, we can decrease the exposure time. This will be done for you automatically if the auto settings are chosen. For example, if we want to acquire one image every 3 minutes for 45 minutes, select the Number of Segments checkbox on the bottom left-hand corner of the Sequence Editor window at a delay of 3 minutes with a number of segments equal to 15. That means we will acquire 15 images with a delay of 3 minutes between each image to equal the total acquisition time of 45 minutes. Click Acquire Sequence to begin imaging. It is important to know that auto settings will override your image capture delay. We want to allow the software the flexibility to obtain maximum sensitivity with the auto feature while preventing it from overriding our set time interval. The default auto settings use a 60-second exposure maximum. However, in the first few captures, we may need more than 1 minute exposure time to obtain the desired count values. Therefore, we suggest changing the max exposure time to the delay time minus 5 seconds to allow for image read time and motor movements. For example, if we want to take an image every 3 minutes, set the auto setting exposure max to 175 seconds. To view and change the auto min-max options, go to Edit on the main menu and choose Preferences. Click over to the Acquisition tab and select Auto Exposure. Change the max exposure time shown in seconds accordingly. In this example, our max time would be 175 seconds. Click Apply when you're done. The software is now ready for acquisition, and you can begin imaging your animals by clicking Acquire Sequence. Label the study appropriately in the Edit Image Labels window and click Save. The software will acquire images for approximately 45 minutes. With up to 2.5% isoflurane, healthy animals can generally be sedated for up to an hour with no issues. But make sure breathing is not labored and consult your animal protocols and veterinary staff if you have additional questions. When finished, don't forget to remove your animals from the instrument and place them back in their respective cages. Now that the software has finished acquisition of the kinetic sequence, we can begin analysis of our data. It is good practice to normalize all images to the same color scale bar. To do this, expand the Image Adjust section of the tool palette and uncheck the individual button. Doing so will allow you to compare all the images in the sequence. The minimum and maximum points on the color scale will incorporate 95% of the highest signal within the entire sequence. Double-check that the signal level is above background noise, 600 counts, and below camera saturation, 60,000 counts. If the signal strength is too low or too high, you will need to reacquire the image using more or less sensitive settings, respectively. After you have checked the counts, switch the units to radians to normalize for changes in acquisition parameters as discussed in the previous in vitro bioluminescence video. By switching the units to radians, we can gauge where our bioluminescence signal was strongest by visual inspection. We can also draw regions of interest to quantify the radiance values and find the time point with highest light output. You may notice that tumors are present in multiple areas of the animal. It is important to establish how you wish to analyze your data. You can either draw ROIs around each lesion or draw an ROI around the entire animal if you are interested in total tumor burden. If you're interested in comparing thoracic versus peritoneal cavities, for instance, we would determine the peaks for each of these regions separately because signal kinetics can differ throughout the mouse depending on diffusion rates, vascularization, and other anatomical structures that alter bioavailability of the substrate, for example, the blood-brain barrier. For this example, we will draw a single ROI over each lesion and we will draw the ROI big enough to capture the entire diffusion pattern of light. Pro tip: to adjust all ROIs at the same time, hold down the control key. Once the ROIs are positioned correctly and the radiance unit is selected in the image window, click the Measure ROI button to bring up the measurement ROI table. Verify that the units in your table are also correct by checking the Measurements Type drop-down menu in the bottom left-hand corner of the ROI Measurements window. If the units are incorrect in either the image window or in the Measurements table, your table will be populated with NA instead of numbers. The table values can be copied and pasted into a spreadsheet program like Excel. Once the ROI values have been imported, find the plateau of bioluminescent light intensity. A recommended method to determine the optimal imaging time is to graph times versus the percentage of max signal. You should image where the light output is 95% of the max or greater. Typically, you will focus on the peak of your curve as a target as it offers the brightest and most consistent signal. In our example, the peak signal for the whole animal ROI is approximately 25 minutes post-injection for both tumor locations. If these two curves did not overlap within the plateau defined by greater than 95% signal intensity, we would have to image these tumor models separately for the most sensitive and quantitatively consistent analysis. Now that you know how to assess the kinetics and how to acquire new images, let's review a few features in the software. To open a batch of images for analysis, you can either click the Browse button or go to File, Browse, from the main menu. This will launch the browser and allow you to easily search for images. Choose the parent folder that contains all the images you wish to view. If there are folders nested inside of the parent folder, the software will search those too. All images will appear and are named by user, date, and time. You will see details that you added when you filled out the Image Labels window, and you will see a preview window on the left. If the images were acquired as part of a sequence, you'll see a green SEQ icon. Clicking the drop-down caret will reveal all the individual images that are part of that sequence, each possessing a red IMG icon. That IMG icon will look slightly different if the image contains saturated pixels. Once you've navigated to the folder you want, you can either load a single image, or hold the Control key and click to select multiple images to be loaded as a group. As an example, we will load a longitudinal study where orthotopic glioblastoma tumors are growing in nude mice. Because the images were loaded as a group, they appear as a sequence on the screen. You can double-click on a single image to open it up individually, or you can X out of the image to return to the sequence. Some buttons in the tool palette will become inactive when in sequence view. If that is the case, simply double-click on a single image and they will become active. The default display is luminescence shown in a rainbow color scale overlaid onto a black and white photograph for anatomical reference. You can choose to show the photograph and luminescence separately, but it is most common to show them overlaid. These images may have been acquired using different settings, so to compare them, convert the units to radiance, and uncheck the individual button in the tool palette. You can adjust your preferences so that images automatically threshold to the same scale when loaded together. Go to Edit, Preferences, and deselect this checkbox. This preference will only be changed for the user that is currently logged in. Now you can adjust the color scale min-max values until it displays the pixels to your liking. Please note that changing the scale bar does not change the quantitation. It only modifies how the luminescence pixels are displayed on the screen. We have loaded the images as a group, converted the units to radiance, which corrects for any differences the images might have in exposure time, binning, or f-stop, and set them to the same scale for comparison. The last steps are to draw and appropriately place your ROIs, generate a table, and export the data into the desired graphing program. From this dataset we can determine that mouse number 1 had the highest detectable metastatic burden and mouse number 5 failed to grow a tumor at all. We now understand what the expected growth of tumors will be without intervention, how to divide animals into cohorts, and which animals may need to be excluded from the study. Using the methods we described, you can monitor the expression of your bioluminescent reporter longitudinally to measure efficacy, localization, and progression of your animal models. We hope you found this video helpful. If you have questions or would like to speak to an application scientist, please contact us. Thank you.